toxicity parameters. Mostly experimental people, they check different kind of analysis with the assay. These are the 5 most popular toxicity assay for the skin sensitization in experimental setup. So this specific software, what they have done, they have taken those 5 different toxicity assays, experimental data for the development of the 5 different models. And those 5 different models are used for the prediction of the future drug. Here also you can provide the SMILES representation of your molecule or your SDF format. And in the output, you will get 5 different prediction for the 5 different analysis. And you will get another important prediction, which is consensus prediction. That means if 2 of your models says skin sensitized, toxic, and 3 says non-toxic. So based on 5 different model score, you will get a consensus model prediction that is it really toxic or not based on 5 different models. So how it looks. So this is the site where you need to provide the SMILES, or you need to draw the structure if you know the structure. And once you click the predict, it will give you all 5 analysis. Is it sensitized or non-sensitized? And finally, you will get a consensus model where you will get the final prediction or the final verdict whether your molecule is skin sensitized or non-sensitized. Along with that, you will get a probability map. Like if you see non-sensitized, this specific portions is good, but this specific section is making a little bit problem. So you need to change this section. It may be more non-sensitized. These 2 are sensitized. If you see this highly green zone, we need to change those specific section by computational method, your computational idea, and then further proceed for the further checking, because these 2 specific drugs are not sensitized for the skin. Now, another web server is SwissADME. If you check a different computational drug discovery literature, the SwissADME is one of the most popular site, where along with the physicochemical property prediction, pharmacokinetic property prediction, they also do drug likeness score and medicinal chemistry readiness, which are another important criteria for the drug discovery. And this is the specific site where you can find this specific web server. Along with the ADME prediction, this web server has a few more drug design tools which are connected together, which you can see if you go through this specific website. And this is the complete scheme of the SwissADME. So once you open this specific link, you will find this specific window. Here you can draw the structure, or you can provide your SMILES representation. Once you click the run, you will get all the predictions, your physicochemical properties, lipophilicity, water solubility, PKPD properties, everything. Now, they also give a bioavailability radar plot. What does it mean? This specific pink shadow area is the ideal for any drug. So these red dots are for your query molecule. Now, if these red dots are within this specific shadow area, that means your drug is very good for the all these admin properties or the drug likeness score. If any one of them is outside, then you need to check this specific property very carefully. For our case, the insaturation has some effect. Here we have taken the aspirin, one of the most common drugs, So this thing has some concern, which we need to check with the different methods. If you want to take all the data for your further study or for your publication or for thesis, you can take in a form of CSV file. Now, when you click this show boiled egg, you will see another specific diagram where y-axis is mostly lipophilicity and the x-axis mostly service accessibility properties. Now, you can see this specific diagram called as a boiled egg diagram. You can see this is an egg. This yellow specific portion is yolk of the egg. And if your molecule is residing in the specific yolk of the egg, for example, our molecule is here, that means your drug is very good possible drug, because this is the ideal zone for any query molecule. So the Swiss ADME has a very good representation of your data, along with different kind of diagram or graphical representation you can find, which you can use for your presentation, you can use for your thesis, as well as your publication purposes. There are another two web server I want to talk about before going to the next level. And, the next one is Xenocyte. Along with the metabolism like CyPreact, this also can predict the metabolism of the specific drug. Along with the metabolism, it also can identify the reaction of the specific drug inside the human body. What kind of reaction it can predict? It can predict like epoxidations, quinone formations, bioactivations, UGT or N-dealkylation formations. So these are the major kind of properties generally we consider to check the different kind of toxicity. This specific Xenocyte web server, these are the very unique features which is not available in the previous web server. So if you are interested to check your molecules for the specific reactions, then you can use this specific web server. And this web server also present a schematic diagram which of the specific portions is responsible for the specific type of toxicity. For quinone formation, you can see the specific benzene portion is the most important section, which is responsible for the quinone formation for the paracetamol. The final one is Protox2. Like previous web server, this specific web server has also 33 models, which developed based on the machine learning models, which is freely available in this specific website. Now, along with the oral toxicity or organ-specific toxicity, this specific web server has two specific most important side, which is it can predict toxicological pathway and toxicity target, which are sometimes very important for the drug design purposes. Along with those, you can also find the oral toxicity and the dose in form of milligram per kg. Why those are important? So if you are handling with the animal studies and you want to know about the specific dose, what kind of dose you want to use for the animals, then this specific dose is very much important, because you can adjust those doses to check your animal studies whether your drug is safe or not. Good thing is, if you provide the drug in the specific web server, they give you very detailed description of everything, what kind of endpoints they are predicting and what kind of value they have. Now, these values are basically prediction probability values. If it's close to 1, that means your prediction is 100% sure. If it's close to 50, there's a chance of 50-50. If it's less than 50, that means there's a chance it's not so much confident predictions. So you should check other software. Here, if you see the green color, that means all the green colors, the dark green colors, that means it's surely those specific parameters are non-toxic. If it's extremely red, that means it's really extremely toxic. For our case, this specific drug is extremely immunotoxic. Except this immunotoxicity, other toxicity are well within the threshold. So basically, we need to go for something to control this immunotoxicity. So we need to work on this structure furthermore so that we can work on this specific immunotoxicity, although other toxicity parameters are well within the threshold limit. Now, these are the other miscellaneous tools, mostly for different purposes we have used. I'm not talking about everyone, because after some time it's more in a monotonous scale. So now what I will do, I will show you some practical demonstration, because I talked about lots of software or web server, but most important thing, everyone wants to know how we need to perform our molecule prediction using those web servers. So let me go to the web servers. Is the web servers visible to everyone? Hello? Is it visible?
Yes, sir.
Okay. Okay. So the first, I will talk about three different web server as a demonstration. The first one is SwissAdmin. This is the link. This is the SwissAdmin. They have lots of different web server. You can perform docking. You can perform different kind of other predictions. You can design your drug. You can parameterize your ligand or proteins for molecular dynamics. But I will talk about SwissAdmin today. So for example, let's see, I will take paracetamol or acetaminophen, which is very common drug. I think everyone know about it. Sorry. So this is the structure of acetaminophen or the paracetamol. So basically, this is the parahydroxyl group where this is the structure of paracetamol or acetaminophen. So you can draw the structure if you know. If you don't know the structure, you can provide the smiles. How to get the smiles? You can go to the PubChem. Under the PubChem, you can, for example, if you write paracetamol or acetaminophen, here they have identify this one. Okay, so acetaminophen because it's a more common name, and you need to go to here. This canonical smiles. You can copy it and paste it here. I know the structure, so let's put it in form of um, smiles. And once you get the smiles, you just run it. Let's wait. Yeah, now you get all the prediction here. You get this specific bioavailability radar plot. You can see this insaturation has a little bit outside of the specific acceptable area, but still it's okay because of other properties are extremely well within the threshold limit. You can see all the properties. Now you can say that I'm not from the pharmacy background. I have no idea about the pharmacology. You don't need to bother about that. You just need to click in the specific question mark. You will get all the properties detail, what a kind of threshold we should follow, what is the threshold limit. Every details are here in close to this specific question mark. Now, if you want to take this data, you just need to click CSV, just download it. You will get your data in form of Excel file or the CSV file. Now you can use it for your presentation, thesis, or anywhere. If you click the show boiled egg, you will get this boiled egg parameter. Here I have used only one drug. If you have more than 100 drugs, obviously it can go different places. In our case, the drug is within the yolk. Please remember, if your drug is within this specific yellow yolk of the egg, then your drug is perfectly well balanced, and you can go for proposing the drug for a specific disease. If it's in this specific gray area, that means your drug is extremely bad. You didn't get anything good. You should check further more compounds. If it's within the white, it's like in the medium section, we need to follow other parameters to check whether our drug is good or not. Now, if you want to learn about all the properties, all the parameters, you should follow this specific paper, this specific Nature paper. They have everything in details, or in the fact or in the help, you can see all the details for the specific drug. Now, coming to the second web server, which is Admit Lab 2.0. Here, we can go for the admit evaluation. You need to come here. You need to go for the get started. For this time, I will take the canonical smiles of the acetaminophen or the paracetamol. Here, you just need to paste it and submit it. Once you submit, each molecule will follow, go through the individual models and predict one by one, and it will give you another radar plot. Here, this specific yellow one is the higher limit, acceptable higher limit, and this specific orange one is the lower limit. For your drug, it should be within this specific limit. And if you see, our blue dots are actually, most of the cases, out of 11 or 12 properties except one are within this specific limit. And the log D is just less, with the acceptable lower threshold limit. And you can see all the physicochemical properties, what we need for the analysis. These are the medicinal chemistry property, and these are the toxic property, and these greens are accepted, yellows are the warning, and the reds are like in the toxicity is there for the specific drug. And like the Swiss ADME, you can also follow those eye marks, where you will get total information about each and individual properties or the toxicities for your query molecules. Like the previous one, you can download all the data in form of CSV file or the Excel file, or you can download as a PDF for your analysis. Now, here, I'm not showing it, just you need to download it in form of that. And the final one, okay, it's ready actually. Let's see this one. So you will get in form of like this as a table. Good thing is you can use this table in your any kind of analysis, and they have given everything in form of metabolism, distribution, absorption, toxicity with a different section. The final one is PKCSM. This is also another similar kind of web server. Here you need to go to the predict. Here, let's use aspirin, which is another popular drug. Good thing is, it's depending on you what kind of parameters you need to check. If you want all admit parameters, you can click here. If you want only distribution, just click distribution. You will get only distribution parameter in your predictions. So you can see each individual models are running for the specific molecules under the distribution parameter. And these are the most commonly used for distribution parameters for any pharmacological study. The volume distribution, the blood-brain barrier permeability, the CNS permeability, and fraction unbound. So you can use those specific values in your study. So these are the three major demonstrations I have shown. You can check each and individual web server for your own study. Now let's get back to the PPT one more time. So there may be a question arise that you have shown 10 or 12 different web server, plus you have shown miscellaneous 5 or 10 web server. So how to choose which is the best web server for my study? The best answers are following. Obviously, we need to identify what we want to predict. If we want to predict eye toxicity, and if I'm taking the heart toxicity prediction web server, obviously it cannot predict the eye toxicity because this specific web server is used for only cardiotoxicity. So you need to check all the models, all the databases, all the web server reporting and the literature. If your specific parameters or specific features is there, then only you can predict. So if you want to predict eye toxicity, obviously you need to check ADMET Lab 2.0, ADMET SAR, PKCSM, or Swiss ADME. You cannot go for the Pred Harch. Next thing is, as I mentioned, you need to check the data sets, models, because maybe you are using a steroid kind of drugs, and they have prepared the models based on benzodiazepines, azoles group of drugs. That means even if you use this web server and predict, there is a possibility that your prediction, maybe you will get some values, maybe you will get some positive or negative sign, but basically your prediction is not reliable. The third point is what kind of prediction you want. Do you want the qualitative prediction? That means toxic, non-toxic, yes or no. You don't need specific values because you just want to report is it toxic or non-toxic. You can go for qualitative model. But if you want quantitative prediction, that means you need a value. How much toxic? What is the real value? Because you need to study further animal studies or you want to do some other test, then you need to check each and individual observer if I can get quantitative value or qualitative value for my specific parameters. And the final point, which we believe, always use more than one web server. Never believe one web server because these are predictions. These are machine learning models. These are artificial intelligence models. So always check at least 2 to 3 web server. If you can use more than that and compare the data, compare the relationship, then it's perfect. Under the conclusion, obviously, as I mentioned previously, drug likeness score or the ADMET properties are the most important properties for the to avoid the late stage failure of the new drug candidate. So if you are in this specific kind of research, along with the docking, molecular dynamics, or any kind of binding energy or computational quantum molecular mechanics studies, always perform ADMET properties along with your other tools. Next, obviously, along with these ADMET properties, we are not only taking less time, we are saving lots of money. Plus, in the United States or European Union, you cannot use animal studies just by like that. There are lots of rules, regulations, beta rules, 3R rules are there where you need lots of regulations before doing any kind of animal studies. That's why even US FDA or different kind of US EPA, they are supporting those kind of computational predictions so that people can save animals for those animal studies. Another thing is, obviously, as I mentioned, never believe in single models. Go for multiple models. Go and then, based on your multiple models prediction, make a consensus models and then report them. And the final thing is build your own QSAR model or machine learning models. It sounds funny, but most of the cases for our own studies, we try to develop our own QSAR or machine learning models and use in our own studies. Because many of the cases, when we design drugs, we found that the existing web server or databases, they don't have those kind of specific drugs. And we know that even if we do predictions, the prediction is not reliable. So in those cases, we need to prepare our own databases, prepare our own models, and prepare our own web servers. So if you know how to build the models, go for it. That's the best options. If you want to know about all the observers in details, follow this specific paper. If you don't have the access, you can contact me. I will happily can share the paper with all of you. You can also follow this specific book completely dedicated for this discussion, where in more in-depth discussions are there. For this specific research, I want to thank USA NSF CREST and EBSCO, those fundings. And thank you so much for your time and attention. I'm open for any kind of questions and discussion. Thank you so much.
Thank you, sir, for this was a very informative speech. And thank you for introducing us to CAD, ADMET, taking drug likeness score. It would probably help us a lot in the future. If anybody has any questions, they can ask, sir. Sir is open to questions. Sir, I actually have a request. I'm Aniruddha, actually.
Yeah.
I have a request. It would be very helpful if we could get a copy of this PPT or something.
Okay.
Or maybe a different version of it.
Okay.
Because this was very informative.
The thing I will do, I will send Dr. Subrahma Gupta and Dr. Maiti the book chapter and the paper, which has a more in-depth discussion about all the web servers, how to use them, where to implement them. Everything is there. Only I cannot share the slides actually, but yeah, I will share the chapter and the review, which has very in-depth discussion. Everything you will get it from there.
Okay, thank you so much, sir.
Yeah, you're welcome.
If anybody has any questions, please ask.
I think anyone can ask anything. I know that the topic is a little bit upbeat because of computational drug discovery process, but if you have any kind of query, it can be anything, even if it's not related with the topic. If you are, if you want to proceed your future research in the MTech or your PhD study in this specific direction, you can ask me anything. Or you can contact me through my email ID, or I have provided the website.
Okay, so we can conclude this session.
Okay.
Yes. Thank you so much, sir. Thank you so much to all the participants for attending this seminar.
Thank you. Thank you so much.
Thank you, sir.